see here, also in Switzerland. And not a little bit technical hitch that we have now overcome.
It's okay, you have to push here with much more power.
That's fine, it's just the slide forward. Pointer was all right, it was just the slide forward. So, just like the scenery is different in Toronto, what I want to do is take a little bit of a different approach here based on all the previous presentations. However, I have to say the previous talk has given an excellent segue for my talk right now. So what you see here is actually, as you can see, this was on the front cover of National Geographic in January 2012 issue. And you can see here that these two are identical sisters. And the reason they are identical, as we all know, is because they have got almost all of their genetic material that is quite commonly shared that they have inherited from their mom and dad. But if you take a closer look at them, they are alike, but not quite alike. You can see that there are some subtle differences in them, so maybe certain shape or features of their face. So why does that happen? And I think one of the reasons that may happen is because even though genetics, which we all inherit, and we have that as a permanent, say, blueprint of our genetic code, actually can be modified or superimposed by epigenetic changes. Epigenetic changes are multiple different types. For today's presentation, I'm going to focus on methylation-related changes. So if you see here, not only the genetic changes that we have, which would be superseded by methylation changes or epigenetic changes, but those changes commonly can actually lead to ultimate changes that are seen at protein. So these changes are important not only during normal development, but also in disease development, such as cancer. Now, one of the fundamental key differences, and sometimes potentially considered to be an advantage of epigenetic changes, is genetic changes are permanent changes. Once you have them, they will be there as long as that cell lives. And other clonal population of that cell or other cells that arise from that original parent cell will always carry those genetic changes. But not so much with the methylation changes. You can see that methylation changes are actually quite responsive and sensitive to a lot of different external and internal environmental or otherwise stimuli, such as diet, changes in diet, exposure to different temperatures, and maybe exposure to radiation or drug treatments. So one of the key advantages of methylation or epigenetic changes is they are reversible in nature. And it's really the sum total effect of genetic and epigenetic that leads to the proteomic or protein-based changes. And earlier in the day today, there was a talk that was addressing some of the proteomic changes or protein changes that are seen with the Kallikrein family genes and how to address them using therapeutic targets. So, as I mentioned, epigenetic is actually a reversible mechanism. It's also heritable in nature. Yet another advantage over proteins, they're not quite heritable. And main three aspects of epigenetic changes that can be studied are DNA methylation, histone modifications, and regulation via non-coding RNAs, like microRNAs. Now, DNA methylation, if you would bear with me, is actually addition of a methyl group at the 5' position of cytosine nucleotides. Let's just think back to our school days just for some basic chemistry here. So this is the cytosine, and this is the 5th position of the cytosine among the ACGT nucleotide code. And you can see the addition of methyl group here that makes it to 5-methyl cytosine. When a cytosine is next to a guanine, which is the CG dinucleotide, if there is a methylation, then that is considered to be CPG. That CPG site is a methylated site. When there is such a concentration of CPG sites, it becomes a CPG island. And CPG islands are enriched regions throughout the DNA that may have certain regions that are enriched with methylation mark, which because of its reversible nature can be either added upon or taken away by exposure to different environmental or dietary stimuli. Now, again, just taking that a little bit further, we know that the epigenetic changes can affect or are important in many different diverse functions, both in normal cell and normal development, but also in general developmental stages such as aging or disease development. And this aberrant or abnormal DNA methylation is a very well recognized mark of cancer. In fact, in today's era of personalized medicine, you can see that there would be likely unique and distinct epigenetic methylation signatures or other types of signatures that can exist for different tumors, and they could serve potentially as biomarkers. And if you can see here, you can see that this is one sort of cartoon of a gene. These are exons. They actually lead to making into DNA message into a transcribed into a messenger RNA, which ultimately will lead to a protein product. You can see these circles here. These are those CPG sites that I was referring to. Some of those during normal cell are methylated, but very sparsely and rather randomly. While enriched regions such as the promoter region will have those CPG sites in a normal environment or normal cell not methylated. However, that changes substantially, and in a cancer cell, there is a concentration of these methylated sites in promoter region, as well as sometimes in the beginning of the gene. And the sum total effect of this methylation is essentially the gene expression is silenced, or there is no expression of the gene. And because of this loss of expression, essentially that could reflect into the protein levels. And the protein levels could be either up or down regulated or completely shut down. So with this in mind, I think what we would all probably agree upon that in the theme that has come out of this meeting, at least for somebody who is not a clinician, is screening is important. But who should be screened and how should be screened? I guess from a biological perspective, I cannot really comment at this point on how it should be screened, but I can probably help with who should be screened by developing different types of biomarkers. So for a prostate cancer biomarker, ideally you would like it to be with appropriate increased sensitivity and specificity. And although in today's current era of potential field of diagnostic or prognostic biomarkers, there are quite a few promising candidates, as shown here, PCA3, tempress-ERK fusion protein, or maybe GSTP1 hypermethylation, as shown here. But no single marker has come forward that has actually been able to replace maybe the current evolving and emerging changing field of Gleason score grading and staging, or at least be able to work with it in a complementary fashion. So there is definitely a need to develop more sensitive and more specific biomarkers. So with this in mind, about 4 years ago, we initiated a collaboration which was primarily led by Alex Zlata in Toronto, and also working with Frank Ricker, who's here in the audience, as well as Maciej Kwiatkowski, and other members of the team, as I've shown here. Quite a few of those are here in the audience today. And we all set out to ask a question. The question was how to generate a really good, efficient panel of biomarkers, which would be representative of genetic, epigenetic, and proteomic analysis of Kallikrein gene family. And we would like to be able to develop this multiparametric biomarker panel and call it a GPRO panel. And why focus on Kallikrein locus? Well, there are several different reasons. One extremely important reason is, of course, we all know Kallikrein, the most famous or popular and well-known Kallikrein gene product is really the PSA protein. And so PSA is encoded by one of the members of Kallikrein gene family. But in the literature, there are several other lines of evidence. Kallikrein 2 serum levels have been shown to be increased in several different Gleason, increased in prostate cancer, and also significantly associated with Gleason grade and stage in prostate cancer patient with an increasing association. Kallikrein 4 transcript levels are also correlated similarly with higher grade and stage. And Kallikrein 11 expression is associated with a less advanced stage and lower Gleason score. And this is just to show you that Kallikrein family contains about 15 different genes. They all encode different proteins, and they're all localized on chromosome 19. Some of them are in head-to-tail kind of orientation. Some are in opposite orientation, head-to-head orientation. So with this, we actually examined what was the overall status of methylated or potentially methylated CPG islands in Kallikrein genes. And we came across that essentially there are about 16 different CPG islands, and they are incorporated in these 8 Kallikrein genes. Of those, we actually wanted to focus our attention on to 3 different kallikreins. Those are Kallikrein 6, because there was evidence in the literature, especially stemming from breast cancer analysis, that methylation of Kallikrein 6 actually contributes significantly to breast cancer progression. And there was also Kallikrein 15 coding region and Kallikrein 10 exon 3 region that was shown to be associated with increased methylation. So the rationale for our project was the epigenetic regulation of kallikreins in prostate cancer is not well understood, although there is fair bit of data about differential regulation, either increased expression or decreased expression of several different kallikreins in cancer in general, but prostate cancer in particular. And we wanted to focus our attention to study methylation as biomarkers because of several advantages. Unlike loss of a signal, where there is some decrease, evidence of methylation is a positive and detectable signal, and it can be used, detection of it can be done using quantitative methods such as real-time PCR, and most importantly, you can detect methylation also in archival or formalin embedded paraffin sections or previous tumors, and that really opens up wealth of different types of studies that can be conducted on these. It can also be easily detected in some other biologically non-invasive type of sources, such as DRE urine samples. And again, as I mentioned before, methylation can actually be subjected to reversible and therefore offers down the road maybe potentially some therapeutic options. So that the aim of our study was to characterize these DNA methylation patterns in 3 specific selected kallikreins, Kallikrein 6, 10, and 15. And I have shown here our experimental approach. We actually performed a genome-wide CpG island array profiling strategy, whereby we took a series of different prostate cancer tumors. These were fresh frozen. We extracted DNA from them, and as you'll see on the next slide, we actually were able to identify distinct regions that were enriched in CpG island, and that also showed progressive increase in methylation. Now, once those putative regions were identified for candidate kallikreins, we then independently verified the methylation status using yet another method called EpiTyper, and I will not go into the details of that. And then we also performed literature review to substantiate our findings. And these 3 candidate genes, sometimes methylation, if it is associated with aging, could be a secondary event or more of a bystander effect. So we wanted to ensure that the candidate genes that we chose to study methylation were actually associated with its effect on biological function. And the way we wanted to do that was by exposing these particular methylated status of candidate genes in different prostate cancer cell lines to a demethylating drug called 5-azar-2-deoxycytidine. And this drug will strip away methylation, and so we would expect that a decrease in methylation should lead to increase in its expression, showing that there is actually a biological link. And then finally, after this discovery phase, we performed a validation of our findings in an independent archival specimen, a series of about 150 prostate cancer patients, using a very sensitive and high throughput assay called methylate assay. Okay, and if you would just bear with me with this slide actually has got a fairly important kind of information on it. Each one of these vertical columns represents one single case. You can see we had about 6 normal cases of prostate tissue, Gleason score 6 cases, Gleason score with predominant pattern 3 and predominant pattern 4, we call them 7A and 7B, and Gleason score 8 cases. And you can see on the left-hand side, these were some of the representative kallikrein genes that were present on this particular array. Now, right off the bat, I will tell you that wherever you see red signal, that means there is increased frequency of methylation. If you see a green signal, that means there is no methylation or low methylation, hypomethylation. And as you can see here, if you look at a couple of these kallikreins, like Kallikrein 12 or 13 or 1, there is hardly any change with respect to methylation in any of the region that is shown here for that particular kallikrein. However, 2 kallikreins stand out, and that is 15 and Kallikrein 10. What's also interesting is not only it allows you to tell which particular region of that gene that is likely to incur methylation, it also shows you, because of the selection of these cases, that if you go from Gleason score 6 to Gleason score 8 cases, there is a progressive increase in this red intensity. In other words, there is a progressive increase in the methylation signal. So based on this, as well as similar kind of analysis for Kallikrein 10, we chose 2 separate regions for those genes. And Kallikrein 6 was actually based on previous published literature. And this is just to give you a fairly basic kind of assessment of how methylite assay is done. It's a very sensitive assay that can be performed on very limited, minute amounts of DNA. And essentially, you use your gene of interest, you detect its levels using this fluorescent label probe, and you compare it with a reference gene. In this case, it was the allorepeat gene. And you calculate a ratio that gives you the actual level in a quantitative measure known as percentage methylated ratio, or PMR. And so the first order of business was to confirm and establish that all the kallikreins that we were looking at for their methylation status were indeed associated with their biological link with re-expression. So this is a representative of 2 different cell lines that actually were tested for Kallikrein 10 and Kallikrein 6. And as you can see, this is the native status of methylation in PC3 cells for Kallikrein 10 methylation after treatment with this particular drug, which stripped away the methylation, not completely, but substantially. You can see that the expression actually increased proportionately. And you can see that that is consistent irrespective of the cell line that we used. Following that, we now wanted to perform validation study. And for validation study, we recruited 150 patients. These were diagnosed between 2007 and 2011 at University Health Network in Toronto. And these patients, their complete set of H&E stain slides were obtained. And Dr. Van der Kwast, who is a key member of our team, who is not here today, but he independently, single-handedly re-reviewed all these slides, and so assigned the score, again, with respect to either a concordant score, or if there were discordances, then we wanted to make sure that we were aware of them. Then DNA was extracted from these slides, and this was modified using a certain chemical step to ensure that we will be able to recognize those methylation marks on the DNA that was extracted. And then we looked at its levels. And this is just to show you a representative section of H&E. We did microdissection. This is the area of prostate cancer, and as he marked here, 3+4, and this is the normal area that was taken from the same prostate cancer patient material. And this is actually the list of these patients, and they were stratified according to Gleason score, pathological stage, the weight of prostate, the average age, surgical margin status, and also the PSA in range. And as you can see, the predominant proportion of patients were in Gleason score 7 category. Now moving on to doing the actual methylation analysis of these matched normal and tumor specimens from these 150 patients, we performed analysis to first of all establish or verify that indeed the methylation levels were increased in tumors compared to normal for all these 3 kallikreins. And they were very significantly increased in the tumor compared to matched normal. When we performed the sensitivity specificity assay using the area under curve and looked at receiver operator curve analysis, you can see that among these 3 kallikreins, only Kallikrein 10 actually showed a substantially better performing sensitivity combined with specificity of 82%, with area under curve being about 0.816. So for our subsequent analysis, we really predominantly focused on Kallikrein 10. And this slide shows the association of Gleason score as well as with pathological stage for methylation status of these 3 different kallikreins. As you can see, there was no significant association with Gleason score. However, for Gleason pathological stage, you could see that there was a significant association, particularly for Kallikrein 10, when compared to PT2 versus PT3A or 3B and 4. And the same thing was true for PT2 compared to PT3 for Kallikrein 6. And so in summary, this is the first study that has examined the methylation of kallikrein genes, or at least candidate kallikrein markers, as biomarkers for PCA. Following the treatment of 5-deoxyazacitidine, we were able to establish the biological connection between methylation levels and the associated expression of those kallikreins in prostate cancer. We also established that the prevalence of Kallikrein 6, 10, and 15 methylation is significantly higher in cancer tissue compared to normal, and particularly Kallikrein 6 and 10 methylation levels and its prevalence were significantly associated with pathological stage, but not with grade. Now, this sort of analysis is the first step. What we would like to do is, of course, expand this analysis and to independently verify our observations in other independent prostate cancer patient populations. And those analyses are currently underway. We are looking at yet another patient population recruited in Toronto, and also we hope to transfer the same kind of strategy to be able to use samples that are recruited through our Swiss collaboration here. And so essentially, what we hope to do is by combining genetic, epigenetic, and proteomic markers, which we have coined a new phrase as GPRO markers, we are hoping that we would be able to come up with a panel of markers that can actually be able to complement, if not maybe perhaps substitute, some of the current histopathologic and other kind of assessment and more established criteria. And again, because epigenetic markers are reversible, there is always a potential that it could offer a potential therapeutic avenue for future treatment of certain subsets of prostate cancers. And with this, what I would like to do is acknowledge people who in my group carried out this work. I always firmly believe that the work is as good as the people you have working with you in my team. And I have been always blessed with having really good students and very young and enthusiastic and keen kind of group of people to work with. One of them, whose work I have shown here primarily, is Ekaterina. She's in the audience here, and you can see her picture here. She's trying to look very studious by wearing some glasses, as you can see. The CPG array profiling data that I showed was carried out by Ken Krohn, another PhD student, and Dr. Vaiju Pete. And these are our Canadian team members. Most of them, with the exception of Diamantis and Theo and Neil, are not here. Cynthia Cook and Lee Hong are also part of our team, and they are in the audience. And we would like to sincerely show our appreciation and thank both Drs. Raker and Kwiatkowski for their contribution to this proposal. And thank you for your attention.
With maybe time for just one short question. Jerry?
Yes. Can you go back to the ROC curves? I was surprised that the best ROC curve had the lowest PMR. It was only 4, so there was actually less excess methylation in the 10s. That translated to a better ROC curve than it did for the other 2, where there was a much higher PMR. So the question is, what does that really say about the concept of methylation? It's relative, of course, but it's not a magnitude, I guess.
No, it's a combination. That's actually a very good point. Methylation is both a qualitative and quantitative phenomena. When you are looking at in this context, you have to put together it's the individual levels of methylation that come from different types of tumor cells. And as we know, all tumor cells are not created equal. So yes, there is a discriminatory value that is being assigned in a most optimal level. So at its best, this type of methylite assay is considered to be a semi-quantitative assay. Now, as you may know, that there are many different newer and more modern technologies of high-throughput screening are coming up, especially with respect to sequencing and digital methylite PCR. And those things will hopefully help to further improve that kind of discrimination value.
Good.
Does that answer your question,
Yeah,
Jerry?
very much so. Thank you.
Well, thank you very much.